Hi, everybody. Welcome to the Research and Innovation Week 3MR competition for the Faculty of Graduate Studies. I hope some of you were able to participate in our 3MT competition that took place in the last hour. We got to learn about some excellent research that's happening at Lakehead University. We now go on to our 3MR competition, which is the 3-minute research. And this is an academic competition that cultivates graduate student presentation and research communication skills and challenges them to describe their research within 3 minutes to a general audience. The competition is open to all graduate students at Lakehead University, and they have to explain their research or scholarly creative activity through a video presentation of up to 3 minutes. We are very excited to announce that Shaw is proud to provide each category winner of Lakehead University's 3MR competition an opportunity to be highlighted in a Shaw Spotlight feature. Each category winner will work with a Shaw Spotlight producer to tell their story and to share their project nationwide. Please note that as a viewer, that if your volume needs to be adjusted, you can do that on your own computers, as each person's volume on their computer is to your own computer. Sorry. I'm going to introduce the judges now. We have Dr. Ronald Harpel, Professor of History, Dr. Chandra Shahi, Dean of Graduate Studies, and Dr. Bhatia Staller, Vice President, Research and Graduate Studies. So I hope everybody is ready to see these wonderful videos that we're going to showcase. Our first person is going to be Ashley Maloney. And Ashley's supervisor is Dr. Ashley Thompson. And Ashley's presentation is entitled Investigation of Variation in Drought-Related Traits Among Yellow Birch Populations.
Climate change is expected to increase the frequency and severity of extreme climate events, including periods of drought, negatively affecting forest health and productivity. Trees have the ability to respond to such disturbances by a change in structure or function. For example, in drought conditions, trees have been shown to change leaf shape or size, branch number, the amount of mass distributed to the roots and shoots, as well as transpiration and photosynthesis rates. Species with broad distributions typically have populations that respond to these climatic stressors differently, or preferably show more resilience under stress. However, in order to have a clear picture of how North American hardwood forests are going to respond to stress, it is crucial to have a strong understanding of the less commonly studied species which play an important role for these ecosystems. Yellow birch, Betula allegheniensis, is a widespread species that inhabits major eastern North American mixed hardwood forests, typically found alongside paper birch, sugar maple, and beech. Yellow birch is economically important as the impressive wood is desired for hardwood flooring, furniture, and cabinets. Arguably more importantly, yellow birch provides food and shelter for a variety of forest dwellers, such as yellow-bellied sapsuckers, squirrels, deer, moose, porcupines, and a host of insects and fungi. Now, despite the importance of yellow birch, current range-wide research is limited for this species. Most of the information we have on yellow birch is based off of research that was conducted in the late 1900s. So, the objective of my research is to investigate variation in functional and structural traits, specifically those related to drought tolerance, as well as climate variables that may be driving this variation. In order to do this, I collected yellow birch seed from 10 populations across the species range, grew them in the greenhouse for 2 years under optimal conditions, and then measured traits such as height, diameter, biomass distribution, carbon isotopes, and a variety of leaf characteristics. This is considered a common garden test, where the greenhouse conditions should allow the seedlings to perform their best. I am just now beginning the data analysis stage, and the preliminary results show significant variation in traits such as height, leaf width, and above ground biomass. My next steps are to investigate whether there are patterns of climate variables, such as mean annual temperature, at seed origin that correlate with this significant variation. With my research, I hope to supplement the understanding of variation within yellow birch as a species and ultimately aid in the conservation and protection of North American hardwood forests under climate change. Thank you.
Thank you, Ashley. Our next video is going to be by Shakira Mohammed. Supervisor is Ashlyn Musquash, and the title is User Perspectives on a Resilience Building App, JoyPop, Qualitative Study.
Advances in technology have opened doors for implementing readily accessible resilience interventions that can now be accessed by the simple touch of a smartphone. My name is Shakira Mohammed, and I'm part of a research team that developed and evaluated a resilience building app called the JoyPop app. Initial evidence supports the effectiveness of the JoyPop app in improving resilience-related outcomes, such as improvements in emotion regulation and depressive symptoms, the more the app was used over a 4-week period. Although this initial evidence shows promise for providing a resilience intervention in a cost-effective way, qualitative research is needed to ensure the usability and utility of a mobile health app, such as the JoyPop app. As such, we explored users' experiences with the JoyPop app and their perspectives on its utility. The JoyPop app was designed to target self-regulation through various features that promote self-reflection and self-awareness. For the current study, we conducted semi-structured interviews with first-year undergraduate students who used the app for 4 weeks. Through inductive content analysis, we found that the Rate My Mood or emotion rating feature, which involves labeling emotions, journaling, which involves self-reflection on thoughts and feelings, breathing exercises such as deep breathing, and the Square Moves Tetris-like game were all features that were used the most often and or considered most helpful. Several themes pertaining to facilitators of app use were identified. Daily email prompts reminding students to use the app helped them establish a sense of routine, and the app served as a reminder for students to really take a moment for themselves to self-monitor and express their emotions. The students also expressed several positive outcomes related to their use of the JoyPop app. They indicated that the JoyPop app increased awareness and understanding of their emotions. It served as a signal to check in on how they were doing and assess how they might improve their mood if needed. It helps students learn emotional control strategies to better regulate their emotions, and it was considered a helpful distraction from the stressors of daily life. In conclusion, the study provides insight into the aspects of the JoyPop app that motivated and benefited users to promote longer-term uptake. The current study speaks to the usability and functionality of the JoyPop app, as both effectiveness and usability are vital to establishing the merit of a mobile health app. Thank you all for listening.
Thank you, Shakira. Our next presenter is George Jovanovich. Supervisor is Dr. Morella Stroik, and the title is A Psychological Interpretation of the Phenomenon of Rights Claiming.
What are human rights and what do they have to do with psychology? What kinds of changes and approaches are required? Good questions. This problem was recognized in October of 2021 by the American Psychological Association in its apology for its role in perpetuating and failing to challenge racism, racial discrimination, and human hierarchy in the United States. The apology recognized psychology's role in perpetuating white Western colonialism and acknowledged how as a science it participated in and disseminated scientific models and approaches rooted in scientific racism when the discipline was first founded. And so there's a desire to be able to do better now that we know better. But how do we do better? Many people point to international conventions such as the Universal Declaration of Human Rights or other kinds of conventions, and this certainly is an important part. But leaving at this stage is limiting. Human rights are not simply legally positivistic codes and regulations. They are fundamentally rooted in cultural experience and the concrete act of rights claiming by minority or otherwise marginalized individuals and groups in society. My research locates human rights in the experience of rights claiming of otherwise marginalized groups. Malcolm X explained how if we truly had a meaningful society, it would not be necessary to pass any more laws. All people would be included as equal participants within society automatically. So human rights, therefore, is tied to the real-world experience of people. reflecting on their lived experience and requesting changes to society to enable their full public participation. My research aims to develop a model of human rights that interprets them as psychologically adaptive responses that human agents, particularly in the Western world, develop to claim or reclaim a space in the community. It's important to be able to understand what goes into rights claiming. Understanding the relationship between all of the variables that go into the phenomenon of rights claiming will help psychologists take a rights-based approach in building the capacities of marginalized individuals and groups, support the healing of rifts in society, and guide the development of rights claiming in a psychologically dignified and adaptive manner. It will help psychologists and all those involved in mental health associated work appreciate the nature of human rights to better include it in their practice. And this direction resonates with the purpose of human rights as described by the chair of the Steering Committee of the Universal Declaration of Human Rights, Eleanor Roosevelt, who said, "Where after all do universal human rights begin? In small places, close to home, so close and so small that they cannot be seen on any maps of the world. Yet they are the world of the individual person, the neighborhood they live in, the school or college they attend, the factory, farm, or office they work. Such are the places where every man, woman, and child seeks equal justice, equal opportunity, equal dignity without discrimination. Unless these rights have meaning there, they have little meaning anywhere." Thank you.
Thank you, George. The next presenter is Olarank Onoredi. Dr. Chingling Dai is the supervisor, and the title of the presentation is Psychological and Morphological Response of Yellow Birch to Changes in Photoperiod Regime, Elevated Carbon Dioxide, and Moisture Regime.
What if I tell you that trees are likely to migrate just like human beings migrate? This past research has that trees are likely to migrate to higher latitude as a result of climate change. However, the success of this migration is dependent on several environmental factors that are not present at the climate envelope. Interactive effect of these environmental factors is not well understood. Hence our decision to research on the interactive effect of photoperiod, carbon dioxide, and soil moisture on the physiological and morphological trait of yellow birch. Why yellow birch? Not much work has been done on yellow birch here in Ontario, hence our decision to work on it. Yellow birch, aside its economic value, is an important component of the forest cover type, which is the sugar maple, the beech, and the yellow birch. It also grows on well-drained soil up to 49.5 degrees northward. Seeds of yellow birch will be collected from big thunder and it will be grown, and the seedlings will be exposed to 2 levels of CO2, which is the 400 ambient and the 1000 elevated. It will be exposed to 4 levels of photoperiod, which ranges from the 48 degree northward, the seed at seed origin, to 58 degree northward, the predicted 10 degree northward shift. The seedling will also be exposed to 2 levels of soil moisture, which is the high and the low. Fertilization will be done accordingly, and this experiment will be carried out at greenhouse in Lakehead University. The experimental design will be a split plot design with carbon dioxide being the main plot, the photoperiod being the subplot, and the soil moisture being the sub-plot. At the end of the first growing season, the tree seedlings will be kept in a working cold storage at -4 degrees in the dark. And to start with the second growing season, the tree seedling will be transplanted into a larger pot where it will be thawed gradually under simulated natural spring condition. And at the end of the whole experiment, the response in the physiology and morphology of growing tree seedling to growing seed treating to photoperiod, carbon dioxide, and soil moisture will be well understood in the context of assisted northward migration of the boreal trees, which we are focusing on yellow birch.
Thank you very much, Laurent. Our next presenter is Laurent Cheret, and the supervisor is Dr. Thiago Oliveira. Title is Improving Transfer Learning with Variational Feature Encoders.
Hello. Today I would like to talk to you about image classification systems. What are those? Well, they belong to the machine learning world and help us identify the content of images. Why are they so important? They are present everywhere. They help us classify diseases, have important roles in autonomous driving, reduce costs for companies by performing tasks such as quality control, and keep people safe. How do they work? They are composed of two parts, the feature extractor and the classifier. The feature extractor is a deep convolution network. Its role is to take an image, extract the features, and represent them as a sequence of numbers. This sequence is then fed to the neural network, which has the role of mapping these numbers to a given class. Well, if they are so great then, what is the problem? They fail where humans don't. By creating adversarial attacks, which are distortions on image that are sometimes imperceptible to the human eye, we can cause significant drops in the accuracy of such systems. And so the question now is, how can we rely on systems that can be so easily fooled? Multiple works are trying to change the feature extractor. By creating more complex mathematical operations in the feature extraction process, we can make convolutional networks more robust to noise. The problem is that those changes are sometimes specific to a kind of distortion and sometimes difficult to implement. But still, the problem is solved, right? Not yet. Transfer learning is a very important technique in the machine learning world. Why? Deep convolutional network and other architecture need lots of data to be able to extract the most important information from an image. And so by using transfer learning, we can transfer the knowledge from one model to the other one. And so the new one can be trained on much less data and still generate pretty good results. Thanks to the advances in technology, public libraries and APIs allow us to use these pre-trained models. That is why we can see so much applications of machine learning in our daily lives. And now the question is, are you going to retrain convolutional networks on millions of images every time new changes are made? We propose another approach. Instead of changing the deep convolutional network, why don't we try to remove the noise from the sequence of numbers that it outputs? We use a simple approach that can be easily implemented thanks to technologies publicly available. We place a feature encoder between the feature extractor and the classifier. Its role? To take the long sequence of numbers and represent it in a smaller size by preserving only the most important information. On three types of unseen distortions, our approach showed significant improvement in comparison to the classical architecture. We also saw a significant improvement in the performance of the model on two highly competitive datasets. We hope that this approach will facilitate the creation of models that are more robust. We leave you a table with the results, as well as a list of benefits. Thank you for listening.
Thank you, Laurent. Thank you, Laurent. Our next presenter is Shasha, and her supervisor is Dr. Wayne Bell and Dr. Matthew Leach. Title is How Biodiversity Stabilizes the Ecosystem Under Climate Change.
Hello everyone, I guess most of you have heard about stability. If not, please listen to my short story. We know stability has a broader importance. The stable ecosystem is more capable to steadily provide us with the clean air and water, the opportunities of hunting, fishing, and skiing. The stability of ecosystem functioning is conserved by biodiversity. Higher biodiversity, higher ecosystem stability. However, nowadays, we are facing with the climate change and it's become even variable. Our forest stability is destroyed. You could see the changes in the lower productivity or great fluctuations. With the rising temperature and shifted precipitation, our trees and forests are more vulnerable to the drought. And even worse, the drought days are extended day by day. Also, our water-stressed trees are more easily to lose their defense ability to other disturbance and more prone to die. For example, in North America, the insect outbreak makes the situation even worse. In the past half-century, the unusual defoliation area is more than 23 million hectares, and the total timber loss from insect is twice than that from fire. Under the background of climate change and large area insect outbreak, we face the big problem. That is whether and how biodiversity stabilize forests. We clearly know that the hope of trees and forests to regulate climate change has no way to be given up. So in my first PhD year, I and Dr. Chen used the plot data which distributed in the bar region from 10 provinces of Canada. We used the data to test forest response and the underlying mechanism. Our results, as you see from the structure equation model, it tells us biodiversity stabilized borough forest meant from the positive contribution of phylogenetic diversity rather than the functional diversity. Phylogenetic diversity regulates the midterm mechanism, the species, or to say the population asynchrony, and it also has direct positive effect on stability. Our results highlight the importance of phylogenetic diversity in maintaining the long-term forest stability and in the condition of insect outbreak. Our findings could provide guidance to forest management and biodiversity conservation. At the end, for everyone, I hope to see more actions to protect our living habitat and our nature forest and our nature friend. Thanks.
Thank you, Shay Shay. Our next presenter is Vera Granko. Supervisor is Dr. Mitchell Albert, and the title of her presentation is Long-Term Pulmonary Changes in Subjects Recovered from COVID-19.
Hello, my name is Vera Granko, and I would like to present long-term pulmonary changes in subjects recovered from COVID-19. I'm sure the virus on my slide does not need an introduction. COVID-19 took more than 36,000 lives, and overall, over 3 million cases were reported up to date. Of course, we know how to fight it now, but we still have more than 2.9 million of survivors. We all know that the COVID is a respiratory infection, which affects lungs a lot. So in order to understand the consequences of the COVID-19, we need to evaluate the lung state. It can be done with X-ray computed tomography. However, both of them involve radiation, so they cannot be done multiple times to evaluate the dynamic changes of the post-COVID-19 lungs. In addition, they provide only structural images. MRI, on the other hand, does not show lungs at all due to the absence of the protons in them. That's where hyperpolarized MRI comes into play. Xenon acts as a contrast agent within lungs, and it is visible for MRI. It is able to provide how the lungs function. In our study, we used hyperpolarized xenon to evaluate the long-term effects of COVID-19 on the survivors and to assess the damage that it could cause to lungs. Volunteers who recovered from COVID-19 were scanned at 3 months after recovery. We acquired nice gas phase hyperpolarized xenon images of the post-COVID patients, but they don't provide us quantitative information on the changes. In order to understand them, we need functional images of the lungs. Let's quickly recap how our lungs works. When we inhale oxygen, it gets into alveoli, where it is transferred to the blood flow. Firstly, it dissolves in the blood-air barrier or lung tissue, and then get into the red blood cells that carries it all over the body. Xenon has the same nature as oxygen, so if we evaluate its transfer to the red blood cells, we will get quantitative information about how the lungs function. We acquired images of the xenon dissolved in the lung tissue and red blood cells for post-COVID and healthy volunteers. As a next step, we calculated ratio of RBC and tissue intensities for each pixel. We obtained maps of ratios and calculated mean values. The mean value of RBC to tissue ratio for post-COVID survivor was significantly lower than for the healthy volunteer. This means that the gas transport is significantly reduced in the lungs of patients who recover from COVID-19 infection. In addition, we compared the ventilation heterogeneity in post-COVID patients and healthy volunteers. Ventilation heterogeneity shows how the inhaled gas is distributed within the lungs. The lower and more uniform it is, the better gas spreads all over the lungs. As may be seen at post-COVID survivor, there is a high heterogeneity index at the lower part of his lungs. This means that oxygen is not getting there. In our future work, we're planning to acquire more data at post-COVID survivors at 3 and 6 months after the full recovery. Here's my acknowledgement, and thank you for your attention.
Thank you, Vera. We're now going to take a break and allow our judges to meet and deliberate over everything that they've just seen. We will come back at 6:45 to share the results of the 3MR presentations. Okay, welcome back, everyone. The judges have met and they have provided us with their recommendations. So I'm going to start with third place. And third place goes to Shakira Mohammed. Title, User Perspectives on a Resilience Building App, Joy Pop Qualitative Study. Congratulations. Second place goes to Laurent Churet. Title, Improving Transfer Learning with Variational Feature Encoders. And our first place winner is George Jovanovich. Title of his presentation, A Psychological Interpretation of the Phenomenon of Rights Claiming. Congratulations, George. I want to thank all presenters. You did a wonderful job. Thank you for the excellent research that's happening at Lakehead University. Thank you to our judges, as well as to our audience for participating in the 3MR competition. I urge everyone to continue to participate in the Lakehead Research and Innovation Week and all the presentations that are to come in the following few days. Have a good evening, drive home safe, take care, bye-bye.